Atomic emission spectroscopy, or AES, an essential technique in analytical chemistry for determining the elemental composition of various samples. AES is a process where electrons of an atom in a sample are excited to higher energy levels by an external energy source. As these excited electrons return to their ground state, they emit light at characteristic wavelengths. This method is primarily used for detecting and measuring the concentration of certain metal ions, such as sodium, potassium, lithium, and calcium, in a sample solution. Let's break down the AES process. Instrumentation of a flame photometer or atomic emission spectroscopy. First, we have our sample solution, which contains the metal ions we wish to analyze. This solution is introduced into a device called the nebulizer, which converts the sample into a fine mist or aerosol. Next, we need a fuel source and air from a compressor. These combine to create a flame, our central component. When the aerosolized sample is introduced into this flame, the metal ions within the sample become excited. Let's see what's exactly happening inside a flame. Desolvation. After entering into the flame, the aerosol droplets are then heated to remove the solvent, leaving behind the solid particles, such as salt and NaCl. Atomization. Now the salt solids converts into the atoms such as sodium and chloride by further increase of heat. Excitation and emission. The free atoms or metal ions within the sample become excited. As the electrons in the atoms return to their ground state, they emit light at characteristic wavelengths unique to each element. For instance, sodium salts dissociate in the heat of the flame, forming sodium ions. These ions are then excited to higher energy states, and as they return to their ground state, they emit light at characteristic wavelengths, which can be detected and analyzed. Now, this emitted light is what we need to capture and analyze. Enter the monochromator, or the filter. This component selects the specific wavelength of light emitted by our target metal ions, filtering out other wavelengths we don't need. The selected light is then focused by a lens onto a detector. The detector measures the intensity of the emitted light, which is directly proportional to the concentration of the metal ions in our sample. Finally, the recorded data helps us quantify the amounts of these metal ions. The recorded data is sent onto a system, computer, which gives the relevant information. Applications of AES. Environmental analysis, detecting trace elements in water and soil. Pharmaceuticals. Analyzing metal impurities in drugs. Food and beverage industry. Measuring metal content in food and drinks. Forensic science. Analyzing samples from crime scenes for elemental composition. Please like, share and subscribe and hit the bell icon for more videos.